Hello everyone, I am Nilakanmani, PhD student working with Dr. Piyush Chonsali and Professor Manu Santhanam. I work on utilization of biomass ash in cementitious binders. Now let us see the equipment isothermal calorimeter, which is used to study any reaction or process which involves enthalpy change. Either it can be physical, chemical or biological reactions which involves enthalpy change. Now in our lab, since we deal with more cementitious binders, we use isothermal calorimeter to measure the heat evolution from cementitious binders. So here we will have a sample and a reference sample. Sample is the one for which we have to measure the heat evolution, and then reference sample which will not have any heat production. So the difference between the sample signal and the reference signal will be received as the output to measure the heat evolution. Isothermal calorimeter, as the name suggests, it is operated at a constant temperature. This high performance calorimeter can be operated at a wide temperature range from 5 to 70 degrees Celsius. The test can be performed on cement paste or cement mortar, and a sample size of 20 ml to 125 ml can be placed inside the channel for measuring the heat evolution. Now let us look at the equipment. It has a sleek design with a robust build. So this is a 8 channel calorimeter, and we have one more 4 channel calorimeter. It has a controller state which is used to maintain the constant temperature, and we have 8 channels inside, and which is used to keep the samples in. So totally we have 8 channels, and we have sensors inside which will capture the heat evolution from the cementitious binders. Then we have a software which is used to operate the equipment. We can set the temperature using this. We can set the target temperature, and we can modify the parameters such as duration, time, and then we can enter the details of the sample as shown here, and then we will be able to view the results while running, and we will be able to generate the reports after once the logging is done. Let us get into the sample preparation part. Here we have measured and kept ordinary Portland cement in the vial and distilled water. So we have measured and kept 50 grams of ordinary Portland cement and 25 grams of distilled water, which makes a 0.5 water to binder ratio. So now we will add the water to the cement, and we have to note down the time the moment we add water to the cement, and we have to properly mix to avoid any lumps. We can even opt for a handheld mixer for proper mixing. Now the mix is steady. After mixing, we have to keep the cement paste within 5 minutes to avoid the loss of early age data. The raw materials for the experiment need to be conditioned at the required temperature for at least 24 hours. After mixing, we will place the sample inside the channel. Now let us look at the application part. The heat evolution results from isothermal calorimetry can be used to measure the reaction kinetics. It can be used to measure the activation energy and influence of temperature and water to cement ratio on the reaction kinetics, and it can be even used to measure the reactivity of alternative cementitious binders like fly ash, slag, biomass ash. And since I work on biomass ash, I have used this instrument to measure the reactivity of biomass ash. I have shown the difference, I have shown the plots here. The plots here show the cumulative heat release and heat flow from different types of biomass ash. Here the graph shows that difference in the reactivity of the biomass ash mainly based on its composition variability, which we can assess using the equipment. Thank you.